Hello guys, today we are going to talk about some wonderful structures which can which evolution can create, and they are carboxysome, chlorosome and magnetosome. So they are really amazing, and at least I felt amazing. Let's see, let comment on what you will feel after learning about this. So first of all, carboxysome. To know about carboxysome, we need to know about a special enzyme which can uh, absorb carbon dioxide or fix carbon dioxide and help in the process of photosynthesis. Photosynthesis requires carbon dioxide, right? So that enzyme is Rubisco. You can see over here written Rubisco, and Rubisco is actually a long short form. So the long form of Rubisco is actually ribulose biphosphate carboxylase oxygenase. Okay, so that's an enzyme which is present in plants also, and any photosynthetic bacteria also for that purpose. But that what the function of that enzyme is that this Rubisco can absorb, or it can fix carbon dioxide in its active sites. It is an enzyme, so it will have active sites obviously. So it can fix carbon dioxide in its active sites and help in the process of photosynthesis. Okay, so when the earth was primitive, means this we are talking about bacteria, so they lived in the primitive earth, and the photosynthetic bacteria had no pressure or no tension, because the primitive earth was not didn't contain oxygen, or the atmosphere didn't contain oxygen. So Rubisco can fix carbon dioxide because it's a carbon dioxide was formed earlier, and therefore the plants were photosynthesizing. But during the process of photosynthesis, oxygen is evolved, right? So as oxygen is evolved during photosynthesis, more and more oxygen started accumulating in the atmosphere. This resulted in the increase in concentration of oxygen in the atmosphere, and the problem arises over here. Rubisco has an active site for carbon dioxide as well as for oxygen, right? So the name only suggests that it is carboxylase oxygenase. So it has active sites for both carbon dioxide and oxygen, and therefore oxygen can as well attach to the active site as the carbon dioxide can, and therefore a competitive inhibition of the attachment of carbon dioxide takes place. And if oxygen attaches to the Rubisco, some very wasteful processes which wastes energy of the organism which is doing that process and occurs, and some useless products are formed. So definitely an organism doesn't want to waste its energy forming an useless product, right? So therefore this type of structures are formed, like in the bacteria carboxysome is formed, and in plants have different adaptations too, which we will study in the later chapters or later videos. And plants have adapted, or plants form C4 plants and CAM plants and all those. So let us study about the carboxysome. So how can carboxysome tackle with this problem? Okay, so to study the structure of carboxysome, here you can see that carboxysome contains a outer envelope. Composed envelope is made up of proteins of the family BMC. Okay, so that is not the topic of the video, but it is actually an outer covering, and inside is packed two types of enzymes, or mainly two types of enzymes, which are Rubisco, as I talked about, and carbonic anhydrase. Okay, so Rubisco's function I already discussed, that it can fix carbon dioxide in its active sites. But carbonic anhydrase, what does it do? It actually converts the carbonic acid, or H2CO3 bicarbonate, bicarbonate into carbon dioxide. So that's a physical process, it occurs actually naturally, but this carbon dioxide, or sorry, this carbonic anhydrase can facilitate that process and cause it to happen even more faster. In fact, this is among the fastest enzymes in the list of enzymes. So that was an added information anyways. So carbonic anhydrase, let me write these points. Carbonic anhydrase anhydrase converts bicarbonates or carbonic acid which is H2CO3 carbonic acid to carbon dioxide or CO2. Okay, so that's the function of carbonic anhydrase. And Rubisco can fix this CO2 and carry on photosynthesis, as I told. Okay, so these are the two enzymes. Now how do they work, means how do they co-work? So what happens is that we know that carbon dioxide can react with water to form this H2CO3, and that actually happens in the protoplasma of the bacteria. Now oxygen doesn't react with water, it is stable. So therefore Rubisco, if it is kept freely in the protoplasma or the cytoplasma of the bacteria, this Rubisco can attach itself to oxygen also, it can attach itself to carbon dioxide also, right? But this Rubisco is bound, or it is surrounded by this envelope or outer structure, outer covering. So therefore neither carbon dioxide freely or neither the oxygen can enter this outer covering and attach to Rubisco. Now what can enter this outer covering is the protoplasma. It can diffusion may occur through this outer covering, and protoplasma may enter this carboxysomes. So if protoplasm enters this carboxysome, protoplasm contains a little amount of this carbonic acid H2CO3. So when this protoplasm enters, this carbonic anhydrase kicks on. So carbonic anhydrase converts the carbonic acid to CO2. Now in this little space, the concentration of CO2 is high and oxygen is null or almost not there, and the CO2 is directly fixed by Rubisco and the photosynthesis is carried on. So it is such a process. Okay, so to just revise it again, the protoplasm enters along with the carbonic acid, which is and this carbonic acid is selected by the carbonic anhydrase and converted into CO2, and since oxygen is not there because it is bounded by outer covering, so oxygen cannot enter, only CO2 is present inside the covering, and this CO2 is fixed by Rubisco and carried on, photosynthesis is carried on. So that is the function of carboxysome, which is obviously present in most of the photosynthesizing bacteria. Okay, now the next one, that is the chlorosome. Now the chlorosome is another structure which is present in the photosynthesizing bacteria, and this chlorosome, what it does, or what the name suggests, chlorosome, let's see what does the name suggest. Name suggests chlorosome. Zome means a structure, as you can see, carboxysome, magnetosome, chlorosome, all are structures, right? And or a membrane bound structure also. Anyways, zome can have such a interpretation, but chloro means that it contains chlorophyll. So chlorosome is a structure which contains chlorophyll. Now what does it do, or where is it present? This type of structures is actually present in the bacteria which can do photosynthesis and is present very deep inside water, or actually 800 feet or more deep inside water. So at that depth inside water, light rarely penetrates that depth. So to do photosynthesis, light is obviously required. So if light is very scarce, then how can this bacteria do photosynthesis? So that's a major challenge for the bacteria, and therefore evolved the structures called the chlorosome, as you can see over here in the picture. So this chlorosome works on the principle that even though light very rarely enters or penetrates that depth in the water, even if one photon of light enters the water, it will utilize it. It will not waste it. So it has very scarcity of light, and it will utilize every bit of it which it gets. So chlorosome works on that principle, and chlorosome, you can see this part. So this part is the chlorosome, and there is the whole structure. So this part is actually made up of chlorophyll, as you can see, and it is arranged in a mesh like manner over here. So now let me talk about antenna molecules. Now antenna molecules are some molecules which surround the reaction center. So what is reaction center? A reaction center is that place or that molecule, final molecule, which takes the energy and then proceeds to do the reaction. But this reaction center is always surrounded by antenna molecules. They perform two processes. So first of all, they actually prevent the reaction center from getting damaged by over high energy molecules, like if the protons are ultraviolet or more gamma rays, X-rays, so they will take the damage, but they will not make the reaction center damaged. So that's one purpose, and another purpose is that they can take on the energy and pass on to the reaction center, and when they pass on, they can pass on very efficiently. So if a structure is so small, only the reaction center is over there, so then the amount of light received will be very less, because all the lights will be all the photons will be passing through the sides, very colloquially speaking. But if the structure is present like this, large antenna molecules are present, right? If large antenna molecules are present surrounding these structures, then obviously the amount of light that will be passed on to the reaction center will be larger, right? Because these antenna molecules will take the light which is passing so much distance away from the reaction center and then pass on to the reaction center. So therefore increase in surface area which will make more utilization of the light which passes that depth into the water. So therefore chlorosome are large antenna molecules, you can say like that, and that is again present in the photosynthesizing bacteria. So that was about chlorosome, and now the interesting part is magnetosome. So let us look at the magnetosome. Let me clear some space over here. Okay, first let's take this one, let's cut it and paste it over here. Now the magnetosome, let me take the picture of the magnetosome too. Now the chlorosome and the carboxysome were surrounded by protein coverings. Okay, so the coverings were made by proteins, and as you know, the bacteria are prokaryotic organisms and they do not contain membrane bound organelles. But this magnetosome is one of the organelles, you can say like that, which is surrounded by a membrane, and that may be the evolution of the first membrane bound organelles in bacteria, and then more membrane bound organelles arise, double membrane bound organelles arise. Anyways, so this magnetosome has a very interesting purpose. It can actually help a bacteria find a perfect spot, or help a aquatic bacteria find a perfect spot in the sea where the concentration of oxygen is just perfect for its survival, or the concentration of oxygen is not even very large or not even very low. So you may ask the question that why we need less amount of oxygen, because if the oxygen concentration is more, that's better, right? But for bacteria, the concentration of oxygen is very much important, because if the oxygen concentration is more, for example, we can say that if the oxygen concentration is more, then the photosynthesis may be hampered, right? So if photosynthesis may be hampered, because Rubisco can fix oxygen too. So for bacteria, we need a perfect concentration of oxygen, which is not too much or not too less. And this type of bacteria, how can they find that space? Or let's say that this is the sea level. So there, as we go deeper into the sea, we will see that the oxygen concentrations fall. Okay, so how can bacteria find a perfect spot? Let's say that this is a spot, this OATZ area, or oxygen anoxygenic zone. So it is between the oxygenic zone and the anoxygenic zone, the perfect spot. How can they find that? That is with the help of magnetosome. So let us understand the relation over here. How does magnetosome help in finding a oxygenic and anoxygenic spot? Let us understand it. So let's say that this is the bacteria, okay, and here we have magnetosome. Magnetosome actually comprises of either magnetite, that is Fe3O4, or greigite, that is Fe3S4. Okay, so these are two components of magnetism. Let's say most of the times there is Fe3O4, and these are actually the ferromagnetic substances. That is, they have magnetic properties when they show large magnetic dipole moment when they are susceptible to external magnetic field. Okay, and even if the magnetic field is removed, they retain a lot of their magnetism. Anyways, this video is not about ferromagnetic substances. So this Fe3O4 can actually respond to the Earth's magnetic field. So now is where the things go a little bit physical, okay, or related to physics. Anyways, so let's say Earth's magnetic field is present everywhere, and if you have studied that Earth's magnetic field composes of two components, that is, one is the vertical component and one is the horizontal component. Okay, so let's say that is the vertical component and the horizontal component. We can divide it into two components, and there is one angle which it makes, obviously, and let's say that is the Earth's magnetic field, BH. Earth's magnetic field B, and there is the vertical component and the horizontal component. Now when this Earth's magnetic field is passing like that, it is present everywhere inside the water, it doesn't care, it will go inside the water. It is present due to the inside dynamo mechanisms in the Earth's core, but that's all different. But the fact is that the Earth's magnetic field is present, and it contains two components. Okay, so because of these magnetosomes, the magnetosomes will try to align themselves with the Earth's magnetic field. So there is not only this one magnetosome, there are plenty of magnetosomes. Let's say like that there, like that there, like that there. Okay, now what will happen is that these magnetosomes will align themselves with the Earth's magnetic field. So what will happen is that they will come to this side. Okay, all will come this side. Now the picture is going dirty. All will come this side, and they will try to, or they will exert a force this side. Sorry, all will come this side and they will exert a force this side. Okay, like this. They will exert a force this side, and this force will create a torque. This is going very, very much physical, and this force will create a torque. If you don't understand it, anyways, you are not studying physics, and it will align this bacteria along the direction of the magnetic field. So what will happen next is that if the bacteria was like that, then the bacteria will be aligned along the direction of the magnetism. How can I show that? I have to draw that in. So the bacteria will be like this, will become like this, and there is the magnetic field of the earth. So due to the magnetosomes, the bacteria aligns itself to the or parallel to the earth's magnetic field. That's the main point I want to describe over here. Now, what is the use of aligning itself parallel to the earth's magnetic field? What do we get by aligning? So that's where this type of that's where the bacteria can understand where is the oxygenic and oxygenic or perfect location for the bacteria. Now this type of bacteria always possess some type of flagella, so they are always motile bacteria. The bacteria which contain magnetosomes are always motile bacteria. We can say that statement. Now let us consider a normal bacteria which doesn't possess a magnetosome. Now it actually moves its flagella and searches for the correct area where it can survive, and therefore it roams about here and there, here and there, here and there, but it is difficult for it to find the correct area, right? I mean, how will it understand which direction to move? So it will just find out, and if its luck is there, then it will find out the correct area. But now for the bacteria which is aligned in the direction of the magnetic field, and therefore it has magnetosomes, they can rotate their flagella and they will always move along one direction, okay, not roam around or wander in any direction possible. So when they are always moving in one direction, the one component of their movement is along the downward direction, as you can see. So they are actually either and depending upon the direction they are moving their flagella, or the direction they are moving their flagella means if they are moving their flagella clockwise direction or counterclockwise direction, they can move either towards the magnetic field like this or away from the magnetic field like this. So they can actually, and if you see the movement, they can actually move either down, or one component of their movement is either down or up. So if they can move down or up, and they do not roam around or they do not move in any direction like those bacteria which do not contain magnetosomes, they can actually find the correct area, because when they are moving through here, let's see this picture. So when they are moving through here, they will actually find the correct area, and if they even cross this correct area, they can move their flagella in the opposite direction and then move up and find the correct area. So they will understand this area is correct. And how do they understand this area is correct? That is more complex than this. That is because of some redox potentials. Understanding redox potentials, when they are in the correct area, the redox potential they understand, and therefore they stop their movement of the bacterial flagella and all those. So hope this is not too much confusing, but if their movement is towards a particular direction, either up or down, then they can easily find the correct area which contains the optimum concentration of oxygen. But if their movement is towards any direction, random direction, then it will be harder for it to find the correct or optimum area where it can survive. Anyways, if it is not in the optimum area, it can survive, but in the optimum area it is obviously better to survive, or the reproduction rate will be better in the optimum area. So this type of movement is actually called magnetotactic aerotactic or magneto-aerotactic movement. So let us write it. So just now what I explained is actually the magneto-aerotactic movement of bacteria, which I hope it is not too much confusing for you now. So magneto-aerotactic movement in bacteria. So that's how what is the use of this this type of magnetosomes. So one who is studying newly about magnetosomes, as I was wondering that what is the use of aligning itself with the Earth's magnetic field? What is the use? They are aligning itself to a particular direction, but this is the use. This is the use which I found out after researching a bit. So hope you liked the video. So that was about the 3 structures, special structures found in special bacteria. If you like the video, please like it and comment whatever you think about it, and meet you in the next video. Okay, bye.